So continuing on to the presentation, with the explanation of the Lesatlier's principle and the blood glucose level is if the blood glucose level is low, then the gluconeogenesis is needed to pursue with creating glucose from pyruvates. However, if the glucose level is very high, then we will then go ahead. The body will then go ahead and utilize glycolysis to break down those extra excess glucose molecules to create pyruvate. Aldolase. Aldolase is one of them is the mechanism that is utilized to drive either glycolysis or gluconeogenesis forward or reverse. This one mechanism is utilized in a sense that this is, and let me show you in a more clear view. This is the picture right here where fructose 1,6 bisphosphate, it has been broken down utilizing fructose bisphosphate aldolase to create dihydroxyacetone phosphate as well as glycerolaldehyd 3 phosphate. So the way that it does, it consists of an enzyme. So um, with any molecule with any enzyme, it utilizes amino acids. So amino acids, if you know, it has a carbon center, it has carboxyl group, it has a amine group, as well as the hydrogen, and then the residue that is at the end, which distinguish each amino acids from another one. So amino acid are monomers that comprised and then builds up what is called polypeptides, and those polypeptides are proteins, and that's what creates the enzymes. So the enzymes are utilized so that way it can speed up the reaction of any biological molecules. So in this sense, this is the picture in the active site. This is where the substrate, the molecules that are being modified or accelerated, or the reactions are being reactions are being done, are held, and then the enzyme is then going to utilize its residues to go ahead and perform its function, because the other part of the amino acids are utilized to link with other amino acids, and only the residues are the one that making any conformational changes or bonds, so on and so forth. So in viewing here, we can see that this enzyme aldolase B is utilized to bind this amine group into this carbon, as well as bind the hydrogen here. Um, bind the hydrogen here, as well as you can see that the hydrogen leaves the base residue in here. um, the hydrogen is being protonated over here. You can see various reaction. You can see the hydrogen leaving the molecules, and here what it does, it creates the this mechanism creates glyceraldehyde 3 phosphate and dihydroacetone phosphate from the use of fructose 1,6 bisphosphate. How is aldolase involved in glycolysis or gluconeogenesis? So aldolase is an enzyme, as I mentioned earlier. It's utilized to break down the first fructose 1,6 bisphosphate to glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. It is also utilized in a way so that way the fructose 1,6 bisphosphate is created utilizing glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. The reason for that is if the fructose 1 bisphosphate breaks down into two of its molecules, those molecules then proceed their glycolysis process to create pyruvates, which can help in the future to synthesize more energy. However, when we have too much of pyruvates, the more creation of the fructose 1,6 bisphosphate breaking down to dihydroxyacetone phosphate and glycerol 3 phosphate, we can utilize those two molecules to combine and form fructose 1,6 bisphosphate to proceed with glyconeogenesis of converting pyruvates into glycolysis. There are different isoforms of aldolase. There's aldolase A, aldolase B, and aldolase C. They all utilize, as I mentioned earlier, they all utilize its residues right here, as shown here, like different aldolase utilize different residues. So aldolase A utilizes tyrosine 363. What it does, it accepts hydrogen. Aldolase A also utilizes lysine 146. The covalent bond which on that it forms stabilizes the intermediate. Also utilizes glutamate 187. Aldolase B utilizes residue of lysine 229 aspartate 33, and it uses skiff-based mechanism in skip-based mechanism where the nucleophilic attack of amine to aldehyde forms a aldehyde is utilized. So that and I can show you in the next step on what the skiff-based mechanism looks like in terms of the molecule changes. Aldolase C, it does utilize residue of glutamate 332 and lysine 71. It forms a salt bridge with the barrel region, creating a surface of aldehydes more negatively charged, so that way it can bind to any acidic molecules. So this is the example that I wanted to show, where aldolase B is utilizing skiff-based formation to create glyceroldehyde. So right here, and then it does that also to create the dihydroxyacetone phosphate. Expression of aldolase isoforms. So where are they expressed at? So aldolase A is expressed at tissue, and it's and on the genetics genetically it's expressed on their chromosomes on chromosome 16. Aldolase B, it is expressed in the liver cells, and it is genetically located on chromosomes 9 of those liver cells. Aldolase C is expressed in the brains, and it is genetically located on chromosome 17. Aldolase B gene regulation. So the the way that the genes are regulated are based on the hepatocyte nuclear factor, HNF. There are three different types, 1, 2, and 3. So the 1 is very strong transactivator, so and then we have 2, which is non-competitive inhibitor, which competitive inhibitor of the side of albumin promoter. Hepatocyte nuclear factor 3 is a strong repressor. So what it is DBP, which is the D side of albumin promoter, is strong transactivator, and CEBP, which is the cytosine adenosine thymine enhancing binding protein, is weak compared to the DBT in certain locations. So they need to function in a very specific location of the hepatic cells to to re- uh, to perform the regulation. So in summary, we went over glycolysis, we went over gluconeogenesis, the differences in terms of the reaction that occurs. The way that these are regulated again is if we have more ATP, if we have more ATP, then it shows that we have more energy, and then so we can go ahead and utilize pyruvates and then conserve those ATPs into pyruvates to create glucose. And again, that also depends on the blood glucose level too. That determines if the reaction should go forward into from glycolysis towards glycolysis, or go backwards towards gluconeogenesis. It also determines if the cell is very high on AMP, which is the adenosine monophosphate. That means that it's very low on energy, so it utilizes glycolysis to create pyruvate, so that way it can go ahead and create more energy in energy through oxidative phosphorylation, um, uh, uh, through aerobic respiration, through aerobic cellular respiration, and utilizes various reactions such as electron transport chain as well as ATP synthase to generate those ATP to create that equilibrium, that homeostasis that the blood glucose level wants. We did go over the purpose of aldolase in glucose in glycolysis, as well as the reversal in terms of gluconeogenesis. We went over a couple isoforms of aldolase, as well as aldolase B hepatocyte nuclear factor. And, if you have any questions, please feel free to let me know in the comments. And then these are the reference that I utilized for the presentation. Thank you. There will be two-part video.